to start this chapter, we started with looking at the building blocks of our nucleic acids, right? The bases. And we looked at nucleosides, and we looked at nucleotides when you had a phosphate there. Then we looked at how those nucleotides form polymers, and they form nucleic acids, and that's our DNA and our RNA. And then we spent some time looking about how that DNA and RNA is kind of can be transcribed and translated into proteins, which is like the language of the physiology of our body, right? The proteins that are made from that messenger RNA do all the work. So in this last part of the chapter here, we want to start looking at what can happen in this to our DNA in terms of mutations. Like, how do mutations occur, and what are the consequences of mutations when things don't go right? So whenever we refer to a mutation is going to be the change in the nucleotide sequence of a DNA molecule. And sometimes this happens just plain old randomly, and others are caused by specific mutagens, which are chemicals that can alter the structure of your DNA. And sometimes there are chemicals, like something you might like here that has a really fancy chemical organic chemistry type name, like we looked at back in the organic chemistry chapters. But sometimes these chemicals are things as simple as like UV light. So if you think of skin cancer, right? Skin cancer often results from overexposure to the sun, and the reason the sun can be so harmful is from the UV light that is in the sun. That's why sunblocks basically block the UV light from penetrating into your cells. Remember back in the organic chapters, we talked about how a lot of sunscreens have a benzene ring. That benzene ring is basically like a blockade for the UV lights that come from the sun so they don't penetrate into your cells. And the whole reason we don't want that to happen is so they don't cause mutations. So these mutations can come in a variety of forms. One of them is called a point mutation. So a point mutation is when you substitute one nucleotide for another. So in this case, a G got converted to a C. So if you're looking at this sequence of DNA, right, a G gets converted to a C. So then the question is, what happens whenever you have that point mutation? Well, the consequence of a point mutation is going to completely depend on where it occurs within your genome, where it occurs within your chromosome, where it occurs within your particular gene of DNA. Is it part of your DNA that leads to the expression of a protein? And if it is, do you actually change the amino acid or not? So there's a lot of variabilities with these different mutations. So point mutations are one type. You can also have deletion mutations, where you actually just have a nucleotide that is lost, and an insertion mutations where you have a nucleotide that is added. These insertion or deletions tend to be pretty severe whenever they occur within a coding region of a gene. Whenever I mean a coding region of a gene, I'm talking about a part of a gene that leads to the production of protein. And the example I always like to use with these insertion or deletion mutations is if you can imagine a sentence with a whole bunch of three-letter words. And here's one. The fat cat ate the rat, right? So the fat cat ate the rat. Now, if you were to just add a letter, right? And so again, each of these are codons, three-letter words, and so on and so forth, right? They're all three-letter words. If you were just to do we'll do a deletion first, and let's just pick a random letter to delete. I'm going to delete the E and the. So now I'm going to rewrite this without the E. So it's going to be TH and then F, and I still have to write it in three-letter words, right? Because that's how our genetic code is read. So the fat cat ate. All right, it's even hard to write sometimes, but I think I got it here. So if we take out the E, right, we end up with, instead of the fat cat ate the rat, we end up with the tet her at, which makes absolutely no sense, right? And the same thing happens to our DNA. Whenever you have an insertion or deletion, it makes no sense, and that means that you're going to have a protein in the end that doesn't make any sense either, right? So that's why insertion or deletions are usually pretty bad mutations if they occur within a protein or a gene reading frame that leads to the expression of a protein. Now, a point mutation, so this is an example of a point mutation. So a point mutation can sometimes be what we refer to as a silent mutation. So this is when if your original DNA, let's say, is CTT, and the point mutation changes that third nucleotide from a T to a C. So instead of CTT, we have CTC. The messenger RNA in the top one, the original is GAA, and the mRNA in the bottom one is GAG. Because of the genetic code chart and the fact that it's redundant, where a lot of times in particular changing that third base in your codon doesn't have any change in the amino acid, both of these lead to the formation of the exact same amino acid, in this case glutamate or glutamic acid. So in this case, the mutation would have no effect. So that's why it's called a silent mutation, because even though there's a mutation, there's no effect on the resulting protein. So there's no effect on the protein, right? So it changes the DNA, but it doesn't affect the protein downstream of it. And again, the protein that is downstream is what actually is going to have the activity or have a particular function. Now, a mutation can also have changes where the amino acid is different. So here's our original CTT. Now we're going to change the second nucleotide, and we're going to make it, instead of CTT, we're going to make it CAT. You can see the mRNAs that are formed. And now in this case, you get two completely different amino acids. One of them is glutamic acid, GLU. The other one's going to be valine. Those are two very different amino acids that have different properties. So up here, right, it says a mutation, right, produces a protein with one different amino acid, can have a small to moderate effect on the overall protein. That's true in some cases, but in other cases, that single amino acid mutation or substitution can be really, really deleterious, have a really bad effect. And one example of that is in hemoglobin. So this example here is actually found in hemoglobin. So this is an exact mutation that occurs in hemoglobin, and is specifically the mutation that causes sickle cell disease. So people who have the sickle cell trait means they have a mutation in their DNA where all of their DNA, the 3 billion base pairs, is all normal, except 1 out of 3 billion is converted from a T to an A in a particular region within their hemoglobin gene. That leads to the formation of a valine amino acid instead of a glutamic acid within their hemoglobin chain. The result of that is a big change in their hemoglobin protein. That hemoglobin protein doesn't fold the correct way, and it ends up causing the red blood cells to form, instead of a red blood cell typically being kind of very round spherical shape, they end up forming a shape like this. It almost looks like a boomerang. And this is called the sickle cell shape. So if you look at an old flag from the Soviet Union, they had a hammer and sickle on there. But this sickle cell shape basically has these really pointed edges to it, and those pointed edges try to go through your capillaries and bloodstreams. They can be very, very painful, and it ends up resulting in those capillaries being ruptured, and eventually those patients frequently die from that sickle cell anemia due to that one little mutation. Another type of mutation that can cause a really big change is if you introduce a premature stop codon, right? So in this case, the mutation basically introduces a stop codon. So now imagine if your protein is supposed to go from amino acid 1 to 1,000, but now you have a mutation that occurs kind of in the middle there where that mutation introduces a stop codon. Now your protein might only go 1 to 500. And you can imagine if the region over there is important in doing stuff, your protein's not going to work as well as it used to. So again, different mutations can have a wide variety of effects, ranging from no effect at all with those silent mutations to very catastrophic effects like what happens if you introduce a stop codon or for instance in the case of hemoglobin. Now, when these mutations can be passed through generations, it's called a genetic disease. So cystic fibrosis is an example of one of literally hundreds of genetic diseases where the parents basically pass that DNA down through their offspring. So this is when you have basically a known trait, and it can be passed on from generation to generation. This is very different than, say, the example I gave earlier where UV light comes in and mutates your DNA. That's something that occurs kind of more randomly and is not passed on from generation to generation, whereas this type of genetic disease is.